Hey, Vince.
Hi, Mario.
Hey, Vince, yeah. I haven't accepted. I think there are some people waiting already, but I thought it's too early to admit others, right?
I think we could already admit them. I don't know.
Yeah.
You want to have a chat?
Sure. Si ano ba? Si Mary Ann?
I'm sending her a message.
Okay. I'll just admit everyone. Ikahan lang. Hello. Kumusta po? I think Anne is here. I think I should make you co-host, and then and to make co-host. Okay, you're both co-hosts now. Hello.
Oh, Anne, nice hair.
This is my pandemic hair.
So Hans
Andy,
is joining us, Anne.
Sorry.
And are you showing slides?
Yes, yes.
Do you want to try share your screen? I made you a co-host, so you can share your slides from your end.
I like your saxophone.
All right, just a second.
So it's 11:30 there, Anne?
Yeah.
What time is it there, Vince?
And 10:30.
10:30 in the morning. Yeah.
Yes.
Mario, right?
Yeah, yes. Nice to meet you, finally.
Nice to meet you. Are you in the US?
Yeah, I'm in Baltimore,
okay.
DC, area.
Okay, I got it.
There it is.
Yeah. You see it well?
Yeah, yeah, very clear.
Cool.
Very clear.
Nice. So we have like 20 more minutes. I think there's about probably 60 registrants as of the last time I checked.
10 of those are for my family.
That's fine. That's good. That's good. Good sign.
Hans may join us. And
oh, really? That's wonderful. I sent him an email actually, just saying hi, and I shared with him the new paper that came out. I will be back. Yeah, I'm just gonna make some tea.
all right,
Okay, go ahead.
All right.
How's the weather there, Vince?
it's not. It's okay. It's a bit cloudy today, but normally.
Okay.
Nice.
We don't have classes or work today.
Yeah, you have this
because of snow, but we're actually inside. So it's really strange.
Okay.
But it didn't snow that much. That's the thing. Yeah.
I mean, it's been snowing in Houston.
Well, in Houston, yeah. Here, yeah, it's bad there. It's bad because they're not used to snow.
Yeah, I know. Yeah. They're not used to snow. They don't know if they use snow tires or something.
That's right. Yeah.
Well, Maryland, you get lots of snow, right?
We get lots of snow. Yeah. Actually, there's more snow last week than today. We were expecting more today, pero yeah. The thing is, it's really very cold and slippery outside. That's probably the reason why they canceled all events today. It's more dangerous.
Normally, we did get lots of heat waves this summer.
Yeah. Oh, okay.
Normally we have heat waves, but been a really mild summer. We've got lots of rain. Our rainwater tanks are full.
Oh, yeah. Wonder. I know Giselle will be attending, and so should Richmond, but they're not here. I actually sent invites to psychology departments like UP, LaSalle, even Southern Luzon Polytechnic University. They said they're attending, and probably
it's quite nice with this Zoom because you get the worldwide audience, right?
Definitely. Yeah, it's a lot of possibilities.
Especially Anne's work, I think it's really at the state of the art, but then it's also too detailed, but it's really to advertise this. And they just got their work published, so it's nice.
Oh yeah, yeah.
I mean,
it's great. It's great. It's really very exciting field, and also because of its connection with active learning, the hippocampus and everything. So it's really great. So when were you together in?
Anne started her PhD with me in 2010. She finished in
2014. Yeah.
She finished in 2014, and she.
Are you in your office? So you go to your office every day?
Yeah, I just want to do some couple of experiments, so I'm in my office at the moment.
But is campus open?
I mean, I usually go to the office anyway, so I could cycle.
Oh well, yeah, that's right. I miss our campus.
So I cycle far, so it gives me a chance to.
Yeah, because we're required to be tested before setting foot on campus.
I mean, we don't have patients here. We're zero.
Yeah, that's right. Yeah.
So
I actually had my first vaccine already, because like here, at least in Maryland, the faculty and staff, university faculty and staff are part of the priority, category ano ba yun? C something. Yeah.
So are you going to record this?
Yeah, also... it's actually, I think, already recording. Kasi the way they set it up, si Giselle and Mon, automatically nagre-record na once you open it. And I don't want to stop it because I might forget. So Richmond will just edit it. So cut it to
the first part.
Yeah.
Also the YouTube link. Who gets to do
that?
I
think Richmond is making that link. We can ask him. Let me just text them. Let me check with them. Baka kasi hindi makapasok because
So are you going? You're going to do the opening remarks, right? So, then I'll just introduce.
I can introduce. Okay, and then we can just co-moderate. You know,
Yeah, okay.
the after her talk, kasi you know, I'm sure people will be writing their questions on chat, and sometimes it's difficult to go back and forth to chat and like talking. So I already open some. I'm writing and. We have about 7 or 8.
So, okay, I can actually admit people.
Can you admit? Yeah, that's good. Any co-host can admit pala. If Giselle will be here, maybe we can ask her to give the opening. I mean, to give a welcome remarks. She's the president. I can introduce her, then you can introduce Anne. Start.
Hi, Hans. So Hans co-supervised Anne's thesis. You are on mute, Mario.
He's also from ANU.
Yeah.
It's great. We have people from all over. We can start in 5 minutes.
If Anne is back.
Yeah.
I'm back.
People will be joining us probably a minute or two before 7 here or 8 in the Philippines.
Giselle's here.
Hello, Press Giselle.
Good morning, Mario, Vince, Mary Ann.
Good morning.
Good evening. So, is our guest speaker here now?
Oh, yes, she is here.
Okay.
Sorry, I was on mute. I said good morning.
Good morning, good evening, Mary Ann.
Yeah, people are still streaming in. We'll start in a minute. Okay. Good morning, everyone, for those in the Philippines and for those here in the US or in Europe. Good evening. Of course, those who are in Australia, like Vince and Hans. Good morning. So welcome to the first ever Research and Expertise Cluster number 2 of PAASE webinar. We're lucky that our President, Dr. Gisela Concepcion is here. By the way, my name is Mario Santo Domingo. I'm the Vice President of PAASE. I would like to ask Dr. Concepcion to give a brief welcome remarks. Giselle?
Thank you, thank you, Mario. Good morning, good evening, very early morning to those in Europe, including to our guest speaker. Thank you so much for being with us. And I'd like us to greet in a very special way former DOH Secretary Manolet Dairit, who is with us. Anyway, this is really a great opportunity to showcase the bright young stars who are doing research in other parts of the world. So I'm very much looking forward to our guest speaker's talk today. But for everyone's information, please be on the lookout for our PAASE webinar series. They're on Fridays. And we have other events and activities every week. So we have PAASE fireside chats, and we have hosted webinars, and we also have other special distinguished lecture guests, including in a month ago, we had a Nobel laureate speak to us. So thank you for this opportunity to speak, Mario, and thank you for organizing this Rec2-led webinar. Good day, everyone.
Thank you, Giselle. So by the way, our research expertise cluster is called Psychology and Neuroscience. So PAASE has about 13 research expertise clusters. So without further ado, let me call on Dr. Vince Daria from Australian National University to introduce our guest speaker. Vince.
Thank you, Mario, and thank you, Giselle. So our guest speaker for today is a postdoc at the Imperial College in London under Professor Simon Schultz. She leads a project investigating circuit changes in the hippocampus with the progression of Alzheimer's disease in mice models. And finished her PhD in Australia. She actually did her PhD in my lab from 2010 to 2014. After her PhD, so she went back to the Philippines for a while, and then she went to Simon Schultz's lab sometime in 2016. She's been with Simon for 4 or 5 years now. Is that correct, Anne? Yeah.
That's right.
And from time to time, we have collaborations, and we have joint projects with our collaborators in Germany. So let's welcome Anne, and I think you can start your talk, Anne. Thank you, Anne.
Thanks, Vince and Mario, for this opportunity to share my research in this webinar. As you know, I'm joining you from London, where it's currently just past midnight. And for those of you who know me, it's way past my bedtime. Let me see. But I'm quite excited to join you, and so hopefully I will stay awake. Oh, just a second. All right, you see my screen now?
Not yet.
Not yet. There you go.
There you go.
Yeah. So this is me. in London. I commute by bike almost anywhere now, actually. With the pandemic, I've avoided public transport for health reasons. And actually, my sense of direction is not very good. In fact, when I find that when I have to go to a new destination, even though I've already studied the map before heading out, I find that I often have to stop by the side of the road and check my maps. And I find that I have to take a certain route several times before I learn it. It's true, isn't it, that finding our way is so crucial to our survival. And perhaps like you, like me, you've wondered, how do we actually know where we are? How do we find our way around? Where is the brain's navigational system, or GPS? In the 1970s, John O'Keefe was sticking electrodes in the thalamic part of the rat brain. But he accidentally missed his target and hit the hippocampus instead. These electrodes are capable of measuring the electrical activity of brain cells. And the mice or the rats he was studying were made to rotate or move in a counterclockwise direction in a rectangular box like the one shown here. And you will notice that the different locations in the box are labeled A, B, C, D, and so on to G. So O'Keefe observed that there were certain cells that fired only when the animal was in certain locations in the environment. So for example, here is a recording from one cell, and this cell in particular fired when the animal was in locations A and B. So from here to H, from this A to H is one lap or one circle around the box. And from here is the start of another lap around the box. And you will see that in the second lap, the mouse this cell continues to fire when the mouse is in the location A. So O'Keefe called this spatial cell a place cell, and the region in its environment where the place cell fired, he called the place field of the cell. Now, we can think of there being multiple place cells in the brain, and each of these cells fire in a different location. So collectively, these cells form a kind of internal map of the environment. And in principle, if we look at the activity of a population of place cells, we could tell where the animal is in its environment. Now, at that time, it was known that the hippocampus had a major role in the encoding, the long-term storage, and the retrieval of episodic memories. So episodic memories are the facts that we know: who, what, when, where. And they knew this because at that time, there was a man by the name of Henry Molaison. He was an epilepsy patient, and he had very bad seizures and blackouts. It's so bad that the neurosurgeon decided to remove his hippocampus. And the immediate effect of this surgery was that the seizures disappeared. And it seemed like the surgery was a success. But eventually it became clear that HM, as he was known then to protect his privacy, did not have long-term memory. He had amnesia. So he could not create new memories. He couldn't remember facts. However, he could do procedures. So his procedural memory was intact. Now we know this is because the part of the brain responsible for procedural memory is different. And this is why a retired mechanic, for example, who loses his hippocampus may not know what year it is or the names of the people around him, but he will remember, or he will know how to change a tire with his eyes closed. Since then, we've also found out that the hippocampus is among the first brain regions to be affected by Alzheimer's disease. Alzheimer's disease, as we know, is a neurodegenerative disease, and its patients suffer from memory loss. Now, the hippocampus proper composed of CA1, CA2, CA3, CA4. They're part of a greater formation in the brain called the hippocampal formation. And part of this region is the dentate gyrus and the entorhinal cortex. And since the 1970s, other spatial cells in the hippocampal formation have been discovered. So let's recall. O'Keefe discovered place cells, and these are cells that show increased firing when the animal is in a particular place in its environment. And this we call the cell's place field. It's important that you understand the two figures to the left here, because it will be important in the other types, in understanding the other types of cells. So imagine a mouse moving in a square box, okay, like we have here. And the trajectory of the mouse, or the path it traverses, is denoted by the black lines in this figure. The green dots are the locations in the box where the cell fired. Okay, now on the left here is what we call a firing rate map. So imagine dividing this square into smaller squares. And for each small square, we count the number of times the cell fired and divide that by the amount of time the mouse spent in that square. And that gives us the firing rate of that cell. So in this figure on the left, the redder the color is, the higher the firing rate. And the bluer it is, the lower the firing rate. Let me just change. Okay, in the 80s, another scientist by the name of Rank discovered head direction cells. Now, can you guess what head direction cells are? You're probably right. You're right if you guess that head direction cells are cells that show increased firing when the animal's head is pointed to a specific direction. So for example, this head direction cell fired at multiple locations in the box, but in all of these locations, there was a singular direction of the animal's head, and it was in the east direction. In 2005, the Mosers, a husband and wife team, they discovered grid cells. These are cells that have periodic firing fields, and we see that in this image here. We see how the firing fields are repeated in like a hexagonal pattern. And it's believed that these cells are involved in measuring distances. So how far an animal has traveled in space, for example. In 2000s, Neil Burgess and others discovered the boundary cells. And these are cells that respond to the presence of a boundary at a certain distance from the animal. So these cells, all of these cells, they're found not just in the CA regions, but also in the entorhinal cortex, in the pre and paracerviculum areas. And altogether, they form a sort of a positioning system in the brain. Now, Of course, the rat brain is different from a human brain, but there are enough similarities in form and in function, so that by studying the rat or the mouse brain, we can gain insight into the human brain. In 2014, O'Keefe and the Mosters actually won the Nobel Prize in Physiology or Medicine for their discoveries of cells that constitute a positioning system in the brain. So you will recall that O'Keefe discovered the place cells and the Mosters discovered the grid cells. So how do we actually record from the hippocampus? In a freely moving mouse, one of the ways this is done is using electrophysiological techniques, where electrodes are inserted into the brain to measure electrical activity. So there are billions of cells in the brain, and each one is connected to some 10,000 other cells. So all these connections can pass some signal into the cell to which they're all connected. And when the signals meet a certain criteria, this causes the cell to fire. So when a cell fires, a positive charge quickly travels from near the body of the cell down its tail, or what's called its axon. So in principle, if you stick a metal electrode in the brain and position it close to a firing cell, you can measure this change in electrical activity. And if you wire this electrode to a speaker, you can hear the chatter, quote unquote, of a firing neuron. So like this. I hope you can hear it. That is the sound of, well, actually, in this case, it's several neurons firing. Now, there are many cells in the brain, so it's important to be able to measure the electrical activity of more than one cell. And this led to the development of multi-electrode arrays. So for example, here we have an array with 16 recording channels. If you count these circles, there are 16, and each unit is each of them is a recording unit. This is a 4-shank multi-electrode array, and each shank is composed of 16 channels, here shown in white. So you have 4 shanks times 16 channels each, and you get 64 channels in total. And this is a system with 16 modules, each module consisting of 64 channels. So 16 times 64, that gives you 1,024 channels. So this becomes sort of a number game. The more channels you have, the more neurons you can record from, and the better. The main advantage of electrophysiological techniques is their high temporal resolution, and this allows us to see fast changes in electrical activity in brain cells. Now, another way to record from the hippocampus is by imaging the cells. But in order to allow the mouse to move freely while we're imaging, the imaging microscope would have to be miniaturized, so small enough so that it can fit into the small head of a mouse. And this is an example of a mouse navigating an open arena with a miniature 2-photon microscope stuck to its head. And on the right here, you see the images that are taken by the microscope. Now, when we image cells, we're not actually measuring the change in the cell's electrical activity directly. So the firing of a cell is accompanied by an influx of calcium ions. So calcium ions entering the cell. And by using calcium indicators. These are fluorescent molecules that respond to the binding of the calcium ions by changing their intensity. We get an indirect report of the cell's activity. That is, we see firing cells literally getting brighter in images. So in the images here, each flashing blob is a firing cell. Now, the disadvantage of this method is that calcium indicators are these, these chemicals, they have a rise time and they have a decay time. So we're not actually able to follow the cell's electrical activity as quickly as we would be if we were measuring it with an electrode. But the advantage is that we can see the cells we're recording from. So if we were, say, imaging for several days or several weeks, we can confidently say that they are in fact the same cells that we are recording from. So with the multi-electrode arrays, any slight movements can introduce changes in the cells that are being recorded from. So it's hard to confidently say that you are actually recording from the same cells. Now, there's GCAMP is a genetically encoded calcium indicator, and it's a very popular one. It's the one I use, for example. And it's a fusion of, here you have a green fluorescent protein. Sorry. You have a green fluorescent protein and a messenger protein, calmodulin, which binds to calcium. And the advantage of using a genetically encoded calcium indicator like GCAMP is that you can specifically tag a certain group of neurons. So you can say, I only want this protein expressed in this group of neurons or in this region of the brain. And this is a very a powerful technique of limiting the number of, or limiting the neurons that you're recording from. Now, miniature microscopes, they're not quite at par yet with the bigger tabletop microscopes. So in general, they have less resolution. They have less imaging depth. You can't go as deep as you can with the tabletop microscopes. Also, they have a much smaller field of view. And so a lot of people like me still prefer to use the tabletop microscopes. Now, so the question is, if the microscope is in place, how do you allow the animal to move? And the answer is to fix the animal. So one of the popular approaches is to head fix the mouse and let it navigate a virtual reality environment like is shown here. So here you have this microscope and you have a mouse which is head fixed and it's running on top of this styrofoam ball. And it's looking at, it's more obvious in the figure here, it's looking at this screen, this virtual reality screen. And there are optical sensors attached to the styrofoam so that the movement of the ball controls what is displayed on the virtual reality screen. So one advantage of these VR systems is that they allow us to manipulate the environment, in ways that are not easily possible in the real world. But one of the key disadvantages of these systems is there's no vestibular input. The mouse is not able to move its head, and there's also lack of sensory feedback. So the mouse is not actually able to touch what it sees or smell or feel, what it sees in the virtual reality screen. Now, an important note to say is that when head-fixed mice are imaged with a 2-photon microscope in this way, that in order to image the hippocampus, a part of the brain actually has to be removed. And this is because, so here is the schematic of the mouse brain. This is the skull, and this is the part of the brain called the cortex. And here you have the hippocampus. The hippocampus is quite deep. And because 2-photon imaging only allows us to go so far, less than 1 micron, we actually have to remove this overlying cortex in order to be able to optically access the hippocampus below. So you might wonder if the mice are okay with this, and the answer is yes. So they survive quite. They survive well. They live on for many more weeks. And there are no adverse effects in their behavior, even though a part of their cortex is missing. Now, just as an aside, virtual reality has actually been useful in observing or recording from humans in order to observe place cells in their hippocampus. So this has only been done in epilepsy patients who already have intracranial electrodes stuck in their brains anyway. So, you know, these are not like healthy humans who volunteered to have metal stuck in their brains. But so these people were made to play a computer game, a taxi computer game where they drove around a virtual city, and by looking at the electrical activity in these electrodes, they were able to observe the same place cell activity that has been observed in mice and in rats. So going back, one of the disadvantages, I must say, with the virtual reality system is that certain parts of or certain characteristics of place cells in this spatial cells are impaired. And so in our lab, we've decided to, or we've chosen to use a different kind of behavioral system. So we use a floating track. It's an arena that floats on air basically, and the mouse is on it and it's head fixed, and we're able to image it in much the same way as others are with their mice and virtual reality systems. And this is relatively new, and until recently, well, until we showed it, the presence of place cells in such an environment has not actually been shown. So we sought to investigate this. So this is an example of an imaging session of a mouse navigating a circular track. You see here there's a water spout. So these mice are, they're water restricted. They don't have water in their cages, but instead we give them a limited amount of water, and a lot of it is given during the behavioral training sessions or during the imaging sessions. Sorry, I'll just play that again. And this motivates them to run. And this light is the laser light that turns on when we're imaging. The floating track system is equipped with a magnet-based tracking system, which allows us to record the position of the mouse, like here. So actually 2-photon calcium imaging is done in the dark, but I installed infrared cameras to take these videos. So here's a mouse navigating an open arena, and you can see the advantage of this real world system in that the mouse has sensory feedback. It can feel the tactile cues we put on the base of the arena. It can whisk, it can smell whatever's on the arena, poop or urine. So again, the question was, can we observe place cells in such a floating real world environment? So first we made sure that the mice could learn to navigate the floating track. So these are images of the circular track and our open field arena. And these are example trajectories. And we can see that they resemble the trajectories that freely moving mice would have in a circular track and in an open arena. We were particularly concerned in the open arena because we wanted the mice to cover the entire area, because this is what's required for place field analysis. And so fortunately, our training protocol was effective. Here we showed the activity of the mice across different sessions. This is for the circular track and this is for the open field. And we can see, so activity is the amount of time the mouse spends running on the track. And we can see that the mouse, the mice on average learn to navigate by session 9. It takes a lot longer though for the open field, and it takes about 15 or so sessions. And we see the same trend in the average speed of the animals. So by session 9, the mice are moving a lot faster than in the early sessions. And in the open field, the average speed doesn't pick up until session 15. So maybe I'll just do a quick run through of the kind of data processing involved in these calcium images. So this is an example. This is an example of a raw video. Well, it's sped up. But here you see in green the GCamp signal. So the flashes are from firing cells, and red is just a static marker. So all the cells in principle express them. And this allows us to, because the GCamp signal it can be different from session to session, depends which cells are firing, but all cells have MRuby, so it's easier for us to identify the cells from one session to another. But you can see the mouse brain moves, and so there's a lot of motion artifact. And so we use the motion correction algorithm built into this system software called Cayman. And you can see how we can get rid of the motion artifact after applying the motion correction algorithm. Now, after this, we identify the cells. So there are so many cells, especially in the bigger your field of view is, the more cells there are. And it's very time consuming to manually make out the cells and their signals. So it all has to be automated. So from these segmented regions of interest, or ROIs, we extract the signals of the cells. So these are the fluctuations in the calcium signal. And well, they're called delta F over F traces. And from their shape, so if you look at the trains, the different trains, knowing what a firing event, what's called an action potential, knowing the profile of that, we can deconvolve these signals and get a measure of the spiking activity, or yes, the timing, basically the onset of the firing when the neurons fired. And then you will recall my explanation earlier of firing rate map. So we calculate the neural event, or okay, I must say that because we don't calibrate our spikes, our signals, we don't actually know how many specific spikes there are to a certain amplitude. So we'll just call them neural events. Okay. So we calculate neural event rate by dividing the total number of spikes or neural events by the amount of time the mouse spent in a particular spatial bin, and that gives us the neural event rate. Okay, so what have we seen in the floating track behavioral system? So we found reliable place cell activity in the circular track. So here we have 5 different cells, and these blue dots represent the firing locations for each cell. And you can see that they're quite well defined. So these are the place fields of these cells. And if you look at the activity of each of these cells per lap, so as the mouse goes through the circular track, sorry, somebody might have to be muted. Okay, so we can see that the firing activity is repeatable across laps. So here we have up to 58 laps. Now, if we put together the neural event rates, rate maps for each of these, for a population of cells, for one mouse, we get something like this. So each row in this plot, or in this figure, is one cell, and the cells have been ordered by according to the location of their maximum event rate. Okay, and we can see that, for example, for this particular mouse, the place fields are distributed across the entire circular tract. Now, actually, we did try one time. I forgot to say, our water reward is randomly given around the circular tract. Once we tried to fix it in a particular location, and we found that a large proportion of the place fields were only for that reward location. So that taught us to randomize the location of the water reward. So these are the place cells pulled from 289 cells. That's 6 fields of views in 3 mice. So when you move the objective in the same mouse brain, you can have a different field of view, and you'd be imaging a different set of cells. Okay, so we see the same distribution across the track. And actually, up here in this panel here are the locations of the fluorescent phosphorescent stickers on the walls. So in green, those are in green, the phosphorescent stickers. And in red are the doors. So this is the inner door in the circular track, and this is the outer door. And if we look at the distribution of place cells around the track, we can see that a lot of the place cells are actually close to these landmarks. So either the doors or the visual landmarks, so the phosphorescent stickers. So this is consistent with what's been published in other papers. If we look at the distribution of the field size, okay, and on average, the field size is about 20 centimeters. So a circular track is about 32 centimeters in diameter. So if you stretch out the track, if you stretch out the circle into a line, it comes out to be 100 centimeters. So 20 centimeters is about 1/5 of that. And so it's quite huge, no? So place cells don't mean a spot in space, but it's an extended location in space. So we also found that the place cells here, as shown in red, sorry, I forgot the legend, they fired more than the non-place cells here shown in blue. Now, when we transferred the mice from one environment to another, from one track to another, we found that the cells almost remapped completely between arenas. So for example, this is place cell 1, and this is its place field in a floating track A. When we transfer it to floating track B, so it's also B is also a circular track, but it's got different visual landmarks and different maybe tactile cues on the floor. We can see that its place field location shifts. And there are other cells, for example, there's cell 2, and we see the same shift in place field location. We see the same thing in cell 3. Well, in cell 3, actually, the cell sort of loses its localized firing field. And this is a phenomenon called global remapping of place cells or place fields. So this is the collated place fields for all the cells in one mouse. I believe it is for one mouse, and they've been arranged here in order of their maximum location in track A. And this is the same cells ordered the same way, but using their place fields in B. And we see that there's a huge difference. No? This diagonal pattern has disappeared, showing that the place fields have shifted completely. And the interesting thing is that when we rearrange the cells according to their place field locations in track B, we see a nice sort of pattern that we had seen earlier. If we look at the place field shift across all the place cells, we don't see a pattern. So it's not possible to predict where the place cells or where the place fields in the new track will be based on their locations in the first track. So we're able to image across several sessions. Here I show our images for 5 or across 5 days. So there are differences because of the movement of the mouse and just some changes in or some movement in our imaging apparatus. So it's important to register the images from day to day. And so here I've shown the registered GCamp images across the days. You will see that they're not identical, and that's because, as I said earlier, the GCamp image will change depending on which cells are active at that particular time. These are the active cells across the days. So for the most part, all of the cells or most of the cells are active across all the sessions. And these are the place cells for across the different sessions. And what happens is that place cells come in and out of this collection. So some place cells only appear in day 1, and some cells only appear in day 3, and so on and so forth. And when we compare the place fields across the sessions, what we see is that they change dynamically over the days. For example, this is cell 143, and this is its place field on day 1. And it's similar. Its place field location on day 3 is similar, but it changes, or sorry, not that, it doesn't change quite a lot for days 4 and 5. But if we look at the cell 197, for example, on days 1 and 3, they're sort of similar, but it changes on day 4 and sort of goes back on day 5. Cell 85 here, we see almost the same location on days 1, 3, 4, and then it sort of shifts on day 5. So the place fields are dynamically changing over the days. So if you look at the ensemble place field maps, if we order them for different days, we will see remapping. So for example, for this row, all the cells are ordered for their place field locations on day 1. And on this row, all the cells are ordered for their place field locations on day 3, and so on and so forth. And we can see that in adjacent images, there is an overlap in the place field locations, but the longer, the farther away in time, the more different, the faster the change in remapping there is. So this is something that is not this is a change that is observed in freely moving mice, but it doesn't change this fast. In fact, in freely moving mice, the fields don't remap quite as much, even over a period of a month. But in virtual reality environments, we see the same sort of thing as in our setup. Now, importantly, we were able to see place cells in our floating open arena. This was something that concerned us because in virtual reality environments, place cells are really only observed in one-dimensional virtual reality, so in circular tracks, if you will. Once people start to use conventional two-dimensional virtual reality environments, they don't see place cells. And the concern was this is due to maybe the lack of vestibular input, because the head is fixed, so the mouse is not able to move the head. But interestingly, we observed place cells in our floating open arena. And this may be because what's required is not vestibular input per se, but a pairing of inputs, sensory inputs. So in our system, we're able to pair motion to visual input to tactile input, and it's probably this pairing of multiple sensory inputs that give rise to place cells in our open arena. Anyway, so these are 4 different cells imaged on 2 different days, 2 days apart. And just like in the circular track case, we see a remapping, the place fields changing across time. And this is the distribution of place cells across the open arena. And here in white, I've drawn the locations of the visual cues on the walls, and on the floor, the tactile cues are shown in these rectangles. And we see that many of the place cells are close to these landmarks again. So close to the tactile cues on the floor and close to the visual cues on the walls. The field size on average is close to 200 centimeters square. And so our open arena is 32.5 centimeters in diameter. So the area is pi r squared. And well, that's the place of field shift. I forgot what I was going to say about that. So in summary, we've seen that spatial cells in the hippocampal formation form a sort of a positioning system in the brain. And we've seen that neural activity can be monitored by measuring current with electrodes, or by detecting photons, or by imaging. In head-fixed mice, place cells can be observed with virtual reality environments, or, as in our case, a floating real-world environment. Now, the question is, why are we interested in imaging the hippocampus of awake behaving mice? And the answer is to look at differences in the brains of normal mice and in the brains of Alzheimer's model mice. So these are mice, Alzheimer's model mice are mice that exhibit or have the characteristics of Alzheimer's, some of the characteristics of Alzheimer's. And we've shown, or we've seen that in these Alzheimer's brains, there are a lot of plaques, which are here shown in methoxyXO4. So these are 2 different Alzheimer's mice models, 5XFAD, which models early onset Alzheimer's. So in humans, this occurs, I think, below the age of 50. And this is another mouse model, APP23, which models the later onset Alzheimer's disease, so with older humans, for example. So we see the GCAMP signal in green, MRUB again in red, and the plaques from the methoxyXO4, which marks plaques in magenta. And this is in vivo, so with the animal alive under the microscope, and this is ex vivo with the brain taken out after the animal was culled. So here in these figures on the right, the plaques appear as white. And these are quite old mice. So you can see that the plaques are everywhere. They're almost everywhere in the brain. I think the APB23 mouse, for example, was almost a year old or over a year old. And this is a video showing the plaque distribution in an old 5XFAD Alzheimer's mouse brain. And you can see a lot of magenta in there. That's the number of plaques. So in humans, these are seen in the brains of Alzheimer's patients, well, after they die and their brains are examined. So I would like to thank my principal investigator, Professor Simon Schultz, and the members of the Neural Coding and Neurodegenerative Diseases Lab, to which I belong. So I am open to questions, if there are any.
Thank you very much.
So much.
Very nice talk, man. Let's see if people could ask their questions on chat. But while they're doing that, I have a couple of questions myself. So Anne, I have a question. So when does the place cell actually activate? Does it need to memorize a certain location in the place? Does it need to memorize some certain location before it gets be recognized as a place cell, or is it there in the moment you place it on the arena?
So it takes time. It's been shown that in the early sessions, the place fields of these place cells can constantly change. There's a sort of a period of time when the connections, if you will, are strengthened. And then after a certain period, the place fields are set for that environment. But if you, so they're quite robust in the sense that if you had, for example, 10 visual cues and you just removed one, you would still see the place field. But if you make considerable changes, then you do see remapping.
Good, thanks. There are no other questions yet. I've got another question actually.
Yes.
So it was very interesting to see that there's a global remapping, and there's also and you've managed to do some housekeeping where you try to differentiate between a visual cell or a reward cell compared to a place cell. So
no, actually, we haven't been able to do that. Say again.
When you randomize the rewards.
Oh, so, yeah, that's right.
The way you've done that, that's kind of housekeeping. But you've also, the visual response, the visual input could also be what we call a visual cell. And depending on the color, for example, a cell could actually react, not necessarily based on the location. How would you differentiate that, for example?
So just a comment, we did not actually look at reward cells. What we saw was that if we fixed the reward location, that a lot of the place cells had place fields in the vicinity of that reward location. So they're not reward tuned in the sense that we didn't look at the correlation of the reward timing and the firing of the cell. So we were only looking at the location of the animal and the firing of the cell. What was the other question? Oh, the visual cell. So your question was whether, how can we say it's not the visual cue that's tuning it? the location.
I mean, if you move the cell from arena A to arena B, and he had the same visual cue, would the same cell fire or something like that?
Well, in that case, it would be hard to say that the tracks are different because the geometries are the same, and we only differentiate them by the visual cues. Do you get what I mean? So the visual cues, the landmarks, are they play an important role in the animal, I guess, differentiating different locations in the environment. And we see that this effect in the fact that the place cells are distributed close to where the landmarks are.
All right, we've got a question from Giselle.
Thanks, Vince, and great talk, Mary. And I just wanted to follow through Vince's question about visual landmarks. And I think I just thought about the primordial reaction of organisms, which is flight or fight. And so I was wondering whether your landmarks could be your stimuli in your field, virtual reality, could elicit a response that would be overt because maybe there's fear or there's something that is in the landmark, and whether there would be a withdrawal, so some kind of a stimulus, is a more active kind of response from the mice. My second question is related to this in terms of therapeutics. I know you're just beginning to study the mechanisms, including that of Alzheimer's, but because I do a little work on dorsal root ganglion neurons from mice, and we also use this similar calcium imaging system. But of course, we're also interested in central nervous system pathologies like Parkinson's. And our first assay is actually an intracranial mouse assay. But anyway, I just wanted to know for my second question, how you plan to move forward in trying to look for drugs or effects of drugs, say on Alzheimer's or other neuropathologies. Thanks, Marianne. Sorry, it's 2 questions.
No worries. So the first question is, for the first question, we don't have, there's nothing, I guess, adverse about our these visual landmarks. They're just stickers that phosphoresce in the dark. There are no electric shocks or nothing like that. We give water rewards when the animals run around the track. I must also say that place fields or place cells have been observed even in paradigms where the environment is completely dark. So even without visible visual cues. The second question, so we are not in a position to actually create or develop the therapeutic side of it, but we are in a position to test what others may develop. Because that any effects at the cellular level we would see, and any effects of that to the behavior of the animal we would see.
So thanks, Mary, and because we do have like candidate compounds from venomous snails. They're called gonidine peptides. So they're neuroactive peptides that the venomous snails use to capture their prey. So there have been studies, a lot of publications in this area of research. Thank you.
Thank you.
Thank you, Giselle. Let me read a question from one of our participants here from Ramil Arciosa. He's from Sultan Kudarat State University in Mindanao, and he works in the area of ethnophysics. His question is, how about the changing of directions? How does change of direction affect place cells? Like, for example, considering a free fall of the mice. So how does change of direction affect place cells? That's basically the question.
So the free fall kind of yeah, but that's actually a very good question. So we have I didn't show it because we weren't able to do it in a lot of mice, but you can look at the place cells when the mouse go in one direction, and the place cells that there are when the mouse goes in the opposite direction. And in other papers, what they've seen is not a complete remapping, but there is a considerable overlap in the place fields. And in fact, what you see is sort of a mirror image of the place fields when the mouse goes in the opposite direction.
Thank you. There's another question here from Angelina Nakaya from Iloilo. She wrote, encoding new experience alter the topology of the co-firing pattern among principal cells. So does encoding new experience alter the topology of the co-firing patterns among principal cells? So what are those changes, if any?
So I'm not too clear about what what principal cells are, but except to say that the cells are the firing pattern across a population of cells can be correlated, and so we may expect to see changes in co-firing patterns with new experience.
Thank you. I have a question because I'm familiar with active learning, and so they always mention the role of hippocampus, in active learning, that when you associate different sensory experiences, then active learning occurs. But I just wonder, diving deep into the role of hippocampus, like if you associate, for example, the smell with a particular place, are there specific patterns that are observed compared to when the association, for example, is tactile? So I wonder about the interaction, of these sensory modalities, and how they're different from each other. Yeah,
that's a good question. And I think what happens is the mouse integrates all these sensory inputs, and that helps create these place fields. And that's actually the advantage of, for example, the floating real world system, is that you have more than one sensory modality, and it's not just like in virtual reality systems, it's not just the display, it's not just a visual input. You have a lot of other sensory inputs to draw from. And for example, in our open arena, we've been able to observe the place cells, unlike in the conventional virtual reality, two-dimensional virtual reality environments. And this, we think, is because of the pairing of these multiple sensory inputs.
It's really interesting. I have a second question. Of course, you've been using the term like global, remapping, so we call that learning. I'm curious about trauma related to place, right? And that's why it may be really difficult to deal with trauma, because of this cross over and the complexity of these associations, right, between the neural connections, but I wonder like how and this may be related to Giselle's question about what's next. How do you, for example, discover, how do you study how to treat, for example, Alzheimer's in this case, which is a similar psychological or physiological condition, because there's so much brain events associated with it. So I just wonder if there are studies that you're familiar with about trauma and place. And
no, I'm not very familiar with that. But so there is such a thing as sphere memory, and then what we're looking at is spatial memory. So they're different kinds of memory, but memory itself is quite rooted in the hippocampus, and so that is something I think that they have in common.
Thanks so much,
Mario. I think I'll follow through. What about amnesia? So you're studying Alzheimer's. What about amnesia? That's a temporary loss of memory, and I'm so sure that then you regain it, but you really lose, a lot of the function. Just curious if you're looking at, amnesia related to trauma. Just curious.
Oh, we're not really looking at amnesia related to trauma, but somebody in my lab is looking at working memory, so short-term memory in the same behavioral system, but she uses a Y maze, and the objective of the mouse is to, the mouse has to remember, so it has to alternate going left, right. And so if its working memory is intact, it should be able to alternate directions. So yeah, nothing related to trauma at the moment.
Thank you. Do we have other questions from the audience?
I could fill in the gap.
Sure.
So the way now I understand it, that the place cells that you're saying is practically a combination of all the other sensories, like a visual cue and other sensory input like smell or something. So it's not just a location. So it could be integrated into a visual cue as well. Have you tried to, like, if you are walking in a city and you see all these buildings and you see this intersection as a visual cue, and you move to another city and you see the same set of buildings, would that place cell light up? Or in your case, if you move a mouse from arena A to arena B, but they have the same visual cue, would the same cells activate?
Are you saying that they have one visual cue in common, but all the others are different?
Exactly, yeah.
I would say, so we haven't done that, but I would say no. there would be complete remapping. Just because as a whole, you know, the collective, the collection of visual cues would be different enough to make the animal think it's a different environment.
But you see what I'm getting at. So if I go as
I see what you're getting at now. It's not just one particular visual cue. It is the combination and the fact that they're all tied to that location in space. You know what I mean? So for example, if the mouse happened to pee in this particular spot in track A, so it's a particular smell, but it's because the smell is in that location.
Yes. There's a question here again from Angelina Nakaya. What happens to old memories when we learn new experiences? Do we have to forget something old to make room?
What gets retained is what gets used. So it's not necessarily that the old memories have to go to make room for the new, but whatever is important to you, whatever you want to remember, that memory has to be accessed over and over again so that the synaptic corrections can be strengthened and that stays there. So in fact, the older the memory, the greater possibility that the synaptic connections are stronger, and so they're there for longer.
Yeah, there's a related question. Is the old information does it get reorganized to integrate new experience?
Yes, it can be. It can be if the experience is related.
But in principle, the older memories will get transferred to the cortex, and
that's right.
Short-term memory is in the hippocampus. But it takes a while to remember older memories if you haven't been accessing it for a long time.
Just wanted to know, Marianne, is there anything known now about how synaptic connections are strengthened? Okay, that would be the basis, say, of memory and learning from the biochemical, level. How does one secure the synaptic connections? What's the biochemical?
So basically, the firing has to be repeated, and so that's why I said the memories that are important to you, they should be accessed constantly.
Giselle, there is this theory called spike timing induced plasticity. So what Anne was saying is that if one cell is firing to another cell, and if they are correlated, the synapse gets stronger. So that's the biology of how memories are stored. In a way, it works because the more you repeat when you memorize, you get to repeat something all over again, and that practically is how memories are strengthened.
Well, I'll tell you at this point that years back, we published a paper in Medical Hypothesis. So it's just a hypothesis. It's wild, but we try to explain it. This is with Ed Padlan. And we said that prions might have a role. They're ubiquitous in all, levels of life. And with constant firing because of repetitive stimulus and experience like learning. Then we looked at the energy of the firing, and we compared it with what is required to misfold or refold the prion protein. And we thought that maybe they would have a role in evolution in securing synaptic transmission, synaptic connections. Anyway, so that's a published hypothesis in the literature.
Thank you, Giselle. It seems like we don't have any more questions. So I know it's already past 1 a.m. where you are. So before we end, do you want to say anything more, like before we end the session? Any last message for everyone about your field and about what you learn from your field?
And not really, but just to say that I'm now processing data from Alzheimer's mice. So stay tuned.
Oh, we look forward to it. So we hope to have another session on your research findings. So Vince, any last words for our audience?
I would like to thank Anne. It was a very great talk. It's nice. I'm very impressed as well. I've visited her lab a couple of times, and I've seen the setup, and I know how difficult these experiments are from our point of view.
Very difficult.
And it takes a lot of perseverance, and well done. Kudos to Anne and your colleagues as well.
Okay, so on behalf of PAASE REC number 2, our REC, we would like to thank you, Dr. Mary Ann Go from Imperial College, Dr. Vince Daria from Australian National University, and Dr. Giselle Concepcion, the president of PAASE, for this very enlightening morning or evening or early morning where you are. So mabuhay po tayong lahat. Thank you very much, and we'll see you again sometime soon. We'll let you know our next PAASE webinar from probably another REC of PAASE. Every 2 weeks we have the webinar, so please stay tuned. Mabuhay, maraming salamat po sa inyong lahat. Thank you.
Bye.
Bye-bye. Salamat po. Many people are thanking you. Very cool. Yeah. Thank you and congratulations.
Can you see the chats, Anne?
Yeah.
Sorry.
Can you see the chats?
The chats.
I can now.
Yeah. People are excited about the field. It's really trailblazing, you know. Very impressive. Thank you all. I will sign off in 30 seconds. You can sleep now.
Yeah. I think
Bye-bye.
my brain went to sleep for a bit earlier.
Bye.
Bye-bye.
Bye, everyone.